Lauren, ready for the semester to begin?
Yeah, it's sad that summer's over, but I'm excited for my classes, especially for genetics.
Me too. The last thing I want to review for genetics is meiosis. Would you be able to help me with that?
Of course, let's go. Remember from last time, in meiosis we go from a 2N parent cell to 4 haploid daughter cells, and there are 2 divisions. That means there are 2 meiosis phases, meiosis 1 and meiosis 2.
Oh yeah, that's right. In meiosis 1, there's a reduction division going from diploid to haploid, and in meiosis 2, there is equational division. Also, meiosis produces genetically variable cells, unlike mitosis.
Yeah, that's the general overview. But once again, I think we need to go into the details.
I've said it once, and I'll say it again, details are the best.
Well, like mitosis, meiosis is preceded by those G1, S, and G2 stages, and interphase where the chromosomes are relaxed.
That's right. Let's start off by talking about meiosis 1, specifically prophase 1. This stage is pretty lengthy and is actually divided into 5 substages: leptotene, zygotene, pachytene, diplotene, and diakinesis.
In leptotene, the chromosomes start to condense and will continue to condense throughout prophase. In zygotene, the homologous chromosomes begin synapsis, where they closely pair up.
That's right. Each homologous pair of synapse chromosomes have 4 chromatids, and in pachytene, the chromosomes become shorter and thicker.
Crossing over is taking place between the zygotene and diplotene substages in prophase 1. Remember, crossing over is when these homologous chromosomes exchange genetic information, almost like a handshake or a high five. Each location where the chromosomes cross is called a chiasma.
Yes, and crossing over creates genetic variation, which we'll talk more about later, and is essential for alignment and separation of homologous chromosomes. In diplotene, the centromeres of the paired chromosomes move apart, but the 2 homologs remain attached at each chiasma.
At the end of prophase 1, the nuclear membrane breaks down and the spindle forms, all in preparation for metaphase 1. So really, prophase 1 is all about condensing and crossing over.
Wow, there was a lot going on in prophase 1. The rest of meiosis 1 sounds way more familiar, just like in mitosis. Metaphase 1 comes after prophase 1. And metaphase is initiated when the homologous pairs of chromosomes align on the metaphase plate.
You got it! Then a microtubule from one spindle pole attaches to one chromosome of a homologous pair, and a microtubule from the other pole attaches to the other member of the pair.
And then comes anaphase 1. This is marked by the separation of the homologous chromosomes. The 2 chromosomes of a homologous pair are pulled toward the opposite poles.
Yeah, and although the homologous chromosomes separate, the sister chromatids remain attached and will travel together.
That is so neat. Then comes telophase 1, where the chromosomes arrive at the spindle poles and the cytoplasm divides.
And that's meiosis 1. The period between meiosis 1 and 2 is called interkinesis, and in some types of cells, during this time, the nuclear membrane reforms around the chromosomes clustered at each pole. The spindle will break down and the chromosomes will relax to the max.
Wow, I almost forgot about interkinesis. I was just too excited to get into prophase 2. In prophase 2, the events of interkinesis are actually reversed, so the chromosomes recondense, the spindles reform, and the nuclear envelope breaks down, again. But unlike prophase 1, there's no crossing over.
That's right. But remember, in interkinesis, some of the cell types have chromosomes that remain condensed, and the spindles do not break down. These cells move directly from cytokinesis into metaphase 2.
And in metaphase 2, the replicated chromosomes line up on the metaphase plate, with sister chromatids facing opposite poles.
Then comes anaphase 2. In this phase, the sister chromatids separate, and the chromatids are pulled to opposite spindle poles. At this point, each chromatid is now a distinct chromosome.
Then comes telophase 2. The chromosomes arrive at the spindle poles, a nuclear envelope reforms around the chromosomes, and the cytoplasm divides, and the product is 4 haploid daughter cells.
Wow, after reviewing meiosis, I feel pretty good. But I think a pop quiz would help, don't you?
My thoughts exactly. Hit me with a question.
Okay. Does crossing over take place in meiosis 1 or 2?
Meiosis 1. And in which phase of Meiosis 1?
That's prophase. In which phase do the homologous pairs of chromosomes align on the plate in the middle of the cell?
That's easy, metaphase 1. And in metaphase 2, the replicated chromosomes line up on the metaphase plate with those sister chromatids facing opposite poles.
Dang, you know your metaphase.
Well, I've got a question for you. In which phase do the chromosomes arrive at the spindle poles and the cytoplasm divides?
I believe that that's telophase 1 and telophase 2. In telophase 2, the nuclear envelope also reforms around the chromosomes.
Finally, in which phase are chromosomes of a homologous pair pulled toward opposite poles?
Easy, anaphase 1 and anaphase 2. And at the end of anaphase 2, each chromatid is a distinct chromosome.
It seems like we know the stages of meiosis 1 and 2.
We sure do. But you mentioned how crossing over is key to genetic variation. What's that about?
Oh yeah, the incredible genetic variation that can occur after cell division can be explained by two processes unique to meiosis: crossing over and random separation of homologous chromosomes, or the law of independent assortment.
Oh yeah, in crossing over, there's an exchange of genetic material between non-sister chromatids. Remember that handshake or high five? Crossing over is the basis for intrachromosomal recombination, which is just the creation of new combinations of alleles on a chromatid.
The second process in meiosis that contributes to genetic variation is the random distribution of chromosomes in anaphase 1 after the random alignment in metaphase 1, which is Mendel's second law, the law of independent assortment.
That's right. Mendel discovered that the combination of traits in offspring of his crosses did not always match the combination of traits in the parent organisms, because after meiosis, each haploid cell contains a mixture of genes from the organism's mother and father. In humans, we have 23 chromosomes. This results in 8.4 million potential combinations of chromosomes from independent assortment alone.
In addition to these processes, another mechanism for genetic variation could be random fertilization. Any sperm can fertilize any ovum.
Wow, what a whirlwind. I think we're now prepared to head into genetics for the semester.
I agree and can't wait to see you all there. Bye for now!